Welcome back to this next video, and in this video we are going to talk about the multiple alleles. So the first thing you need to understand is what is an allele. Now allele is a form of gene which code for one possible outcome of a phenotype. This is how we define an allele, that allele is a form of gene which code for one possible outcome of a phenotype. Let me give you an example of this. For example, in Mendel P investigation, what he found was that there is a gene which determines the color of the pea pod. Now, one form of this gene, like one allele of this particular gene, it creates yellow pods, and the other form of this gene, or you can say the other form of this allele, that create the green pods. This means that there is one gene which is responsible for the color of the pod, but this one particular gene have got two alleles, and these two alleles can actually code for two possible phenotypes. In this particular case, whether they can create the yellow pods or they can create the green pods. Now this means that one allele of this gene will code for yellow pod, and the other form of this gene that will code for the green pods. Therefore, we define the allele as the form of gene which code for one possible outcome of a phenotype. Now, there are two possible phenotypes, and one allele is actually coding for one possible outcome of a phenotype, whether that will be a yellow pod or that will be a green pod. Now, when the gene for one trait, they have got two alleles, and the gene the alleles, they follow the Mendel law of dominance, and there will be three possible genotypes and there will be two possible phenotypes, and the two possible phenotypes, one will be the dominant phenotype and the other one will be the recessive phenotype. We are talking about three possible genotypes because you can have two homozygous condition and you can have one heterozygous condition. Therefore, when there is one gene and they have got two alleles but they follow the Mendel law of dominance, there will be three genotypes but there will be two phenotypes, because the dominant allele will be representing itself in the homozygous as well as in the heterozygous condition, and the recessive one will be expressing itself only in the recessive condition. Therefore, we say that there will be three possible genotypes and there will be two possible phenotypes. Now, when there is one gene and there are two alleles and you are actually having three possible phenotypes, now this means that these two alleles, they are not following the law of dominance, because if they were following the law of dominance, there will be only two possible phenotypes for two alleles. But as we are getting two alleles for one gene, and those two alleles, they are coding for three possible outcomes, you are actually talking about the incomplete dominance or the codominance. I have detailed video on the incomplete dominance and the codominance, and I'll share the link in the description. Now, if there are four or more possible phenotypes for a trait, you have got a trait and there are four or more possible phenotypes for that particular trait. Now that simply means that there will be more than two alleles for that trait, because with two alleles you cannot have four or more possible phenotypes. So this means that if for a trait, if there are four or more possible phenotypes, then more than two alleles for that trait must exist in the population. I'll stress on this. There will be more than two alleles for this trait in the population, not in individual. So when this is the condition, that there are four or more possible phenotypes and you have got more than two alleles, we call this particular condition as the multiple alleles. Now, when you talk about the multiple alleles, as I've told you, there may be multiple alleles within the population, but individuals have only two of those alleles. When you talk about the population, you can have more than two alleles in a population. But when you talk about individual, if you talk about an individual will have only two of the alleles that are present in the population. Now, why this is the case? Because when you talk about the individuals, we have only two biological parents. Now, we inherit half of our genes. That means we inherit half of our alleles from the mother. and the other half from our father. So if there are more than 2 alleles in a population, an individual will have only 2 of those alleles. It cannot have all of the alleles that are present in the population, because we are inheriting 1 allele from our mother and 1 allele from our father. Let me give you an example of the multiple allele. Now, one of the excellent examples of the multiple allele is the inheritance of the human blood type. So when you talk about the human blood type, there are 4 possible phenotypes. Like you can have the type A blood, you can have the type B blood, you can have ABO blood, or you can have the O type of the blood. That means that the human blood type that can have 4 possible phenotypes, the A, B, AB, or the O. Now when you talk about the human blood type, there are 4 possible phenotypes, and there are 3 alleles for the gene that determines the blood type. Now if you can see over here, we are having more than 2 alleles, therefore, and we are having 4 or more possible 4 phenotypes. That means we are talking about the multiple alleles. So when you talk about the gene that determine the blood type in the human beings, that particular gene have got 3 alleles. Now remember, you have just 2 of the 3 alleles in your genotype. In the population, we have got 3 alleles, but in a single individual, only 2 of the 3 allele will be present, because one will be you will be getting 1 allele from the mother, and you will be getting 1 allele from the father. Now, what are these alleles? When you talk about the alleles, there are 3 alleles for the gene that determine the blood type. Now, one of the allele, that is the type A allele, and that codes for the type A blood. The other allele is the type B allele, which code for the type B blood. And the third one is the allele I, which code for the type O blood. If you look at these symbols over here for these alleles, you can actually see that the with allele A and allele B, we are actually writing this capital I over here. Now this means that the this capital I simply means that the if you look at these symbols, let me show you them. If you talk about these symbols, now these symbols says that the allele O is recessive to the is recessive for the allele A and B, because the we are actually expressing the allele A and B in capital letters, and we are expressing the allele I in the allele for the type O in the small letters. That means that the allele A and B, they are dominant over the allele I. Now, when you have got 3 alleles, as in this particular case, we have got the allele A, the allele B, and the allele 3, you can actually have a higher number of the possible combination in creating a genotype. Now, when you talk about the blood group, you can actually have 6 different genotypes. Now, if you talk about this one, if you talk about the type A blood, the type A blood can be represented by the capital I over A, and this A in the homozygous condition, or the type A can express itself in the heterozygous condition, because the allele A that is dominant over the allele I. That means that when the allele A comes with the allele I, the type of the blood, that will be the type A blood. Similarly, when you talk about the allele B or the type B blood, type B blood can express itself in the homozygous condition as well as in the heterozygous condition, because the allele B that is dominant over the allele I. If you talk about this table, from this table you can actually deduce some of the conclusions. Now, one of the conclusions you can deduce from this table is that there are 6 different genotypes. You can see them. It's 1, 2, 3, 4, 5, and 6. And there are 4 phenotypes. You can have the type A blood, the type B blood, the type AB blood, and the type O blood. So you are having 4 phenotypes, and you are having the 6 different genotypes. When you talk about the type A and B blood, they can express themselves in both homozygous and in the heterozygous condition. As you can see over here, the type A blood and the type B blood can express themselves in the homozygous as well as in the heterozygous condition, because the allele A is dominant over the allele I, the allele B that is dominant over the allele B that is dominant over the allele I. Now the allele O, it can only express itself in the recessive condition, or you can say it can only express itself in the homozygous condition. If you talk about the AB blood, like the allele, the type AB blood, it is having the allele A and it is having the allele B as well. Now the allele A is not dominant over the allele B, neither the allele B is dominant over the allele A. Now this means that when this is the condition, both of the allele, they are going to express themselves. Therefore we say that the type AB blood is actually an example of the codominance. Let us talk about the blood groups in a little bit detail, and let's talk about the ABO antigens. When you talk about the ABO antigens, the first thing you need to understand is the precursors for the ABO antigen. And one of the essential precursor for the ABO antigen is known as the H antigen. Now this H antigen, or the locus for this H antigen, is present on the chromosome number 19. It have got 3 exons, and these H antigen are actually expressed on the surface of the red blood cell. So this H antigen is actually acting as a precursor for the ABO blood group antigens. Now, if you look at the nature of this H antigen, this is actually a carbohydrate sequence, and this carbohydrate sequence is attached to the proteins which are present on the surface of the RBCs. Like there are proteins on the surface of the RBCs, and this H antigen is actually attaching to those particular proteins which are present on the surface of the red blood cell. If you talk about the ABO locus, the ABO locus means the gene that is present, that is determining the blood type, whether that is the A, that is the B, or that is the O, or that is the AB. So when you talk about the ABO locus, it is located on the chromosome number 9. It have got 7 exons. This whole locus have got 7 exons, and exon number 7, that is the largest one. And as I've told you that the ABO locus has 3 main alleles. One is the allele A, the other one is the allele B, or the third one is the allele O, or you can simply say the allele I over here. Now, if you look at the surface of the red blood cell, as I've told you, for example, this is a red blood cell of an individual having the O type of the blood. If you look at the sequence of the H antigen, as I've told you that H antigen is actually a carbohydrate sequence. So what is the sequence of this H antigen? The sequence of this H antigen is that you have got a fucose, which is attached to a galactose, which is attached to an n-acetylglucoseamine, which is attached to a galactose. So this is the sequence of the H antigen. Fucose attached to galactose, attached to n-acetylglucoseamine, and then a galactose. And this particular fucose over here, that is attached to the proteins which are present on the surface of the red blood cell. So if this is a red blood cell, it have got this particular protein on its surface. So the H antigen is going to attach to this particular protein. So this is actually the red blood cell of a person having the O type of the blood. We will come to this O type of the blood in the end when we end the discussion on the ABO antigen. First we are going to discuss about the type A blood. Say for example, this is the red blood cell of an individual which is having the A type of the blood. Now this simply means that if you are talking about a person having the A type of the blood, this means that that particular individual will have the allele A. That will be carrying the allele A. Out of the 3 alleles, the allele A, B and I, this particular individual who have type A blood will be having the allele A. So if this is the red blood cell of a person having the allele A, let us discuss this one. This is the red blood cell, and this is the protein present on its surface, and this H antigen is attached over here. Keep in mind one thing, that this H antigen is present on all of the RBCs of all of the blood type. It is present on the RBCs of the individual having the O type of the blood, individual having the A type of the blood, individual having the B type of the blood, or individual having the AB type of the blood. So it is present in all of the RBCs. So if you look at over here, this is the RBC of a person having the A blood. A protein is present over here. So the H antigen, like fucose, the galactose, the n-acetylglucoseamine, and then agalactose. I'm actually using the abbreviation over here. So this F is for fucose, this gal is for galactose, this NAGA is for the n-acetylglucoseamine, and this GAL is for the galactose over here. Now, if you talk about this allele A, this means we are talking about the gene. And this allele A is actually coding for a protein which is known as a glycosyl transferase. This glycosyl transferase is actually an enzyme. So when the allele A, it make this enzyme glycosyl transferase, this glycosyl transferase is going to change the nature of the H antigen which is present on the surface of the red blood cell of the A individual. What this glycosyl transferase is going to do is that it is going to attach another carbohydrate moiety, which is the n-acetylgalactoseamine, to this galactose of the H antigen. It is not going to attach the n-acetylgalactoseamine to this galactose. This is going to attach an n-acetylgalactoseamine to the to this particular galactose of the H antigen. So if you look at this one, the glycosyl transferase, which is encoded by the allele A, is actually changing the nature of the H antigen. Therefore, we call this particular sequence, like the fucose, the galactose, the n-acetylglucoseamine, the galactose, and an n-acetylgalactoseamine attached to this galactose, is the A type of the blood. If you talk about the other type of the blood, say for example, we have got the B type of the blood. Now this means, say for example, this is the red blood cell of an individual having the B type of the blood. This is the protein, and this is the H antigen, the fucose, the galactose, the n-acetylglucoseamine, and the galactose over here. Now this allele B, again, this is a gene, but we are talking about a different allele. This allele B also codes for a glycosyl transferase, but this glycosyl transferase is a different glycosyl transferase. What this glycosyl transferase do is that instead of attaching the n-acetylgalactoseamine, it attach a D-galactose to this particular galactose of the H antigen, again changing the nature of the H antigen. And when this particular H antigen is present on the surface of the red blood cell, we call this is the B type of the blood. When you talk about the allele O, in the allele O, what you are going to see is that let me show you this. If you talk about the allele O over here, if this is the individual having the O type of the blood, if you look at the RBC of that particular individual, it is having this H antigen, like the fucose, the galactose, the n-acetylglucoseamine, and the galactose. What this allele O is doing, that the allele O, it have got a deletion at the exon 6. And when there is a deletion at the exon 6, because of this particular deletion, the enzyme that is coded by the allele O, it loses its activity. Or you can simply say that the glycosyl transferase that is encoded by the allele O, that is an inactive glycosyl transferase. And when the enzyme loses its activity, this results in H antigen remaining unchanged in case of the O type of the blood. Like if you look at over here, the H antigen present on the O type of the individual, that is not changing. The reason is that the enzyme that is coded by the allele O, that have lost its enzymatic activity because of a particular mutation which is known as the frameshift mutation. And when this mutation leads to the inactivity of the enzyme, the H antigen which is present on the red blood cell of the O individual, that remain unchanged. This term O in biology usually refers to the absence of something, and in this particular case, the thing that is absent, that is the activity of the enzyme. So this O is actually for the absence of something. So when the enzyme is not active, that means that the H antigen which is present on the surface of the O type of the individual, that will remain unchanged. That is not going to change. When you talk about the individual having the AB blood type, like there are individuals which are having the AB blood type. Now this means that the individual whose blood type is AB, that means that they have got the allele A as well as the allele B in their genome. They have got both of them. They have got allele A and they have got allele B. Now, what this allele A and B are doing, of course, they are coding for functional enzymes. They are coding for functional glycosyltransferase. So, some of the H antigen, which are present on the surface of the red blood cells, that are changed by the allele A by adding an n-acetylglucoseamine to this particular galactose, and some of the H antigen are changed by the glycosyltransferase coded by the allele B by adding a galactose over here. So when you talk about the individual, the AB individual, they will be having both of these two type of the H antigen present on their surface. Some of the H antigen will be changed by the allele A, some of the antigen will be changed by the allele B. So is both of them, they are expressing themselves. This means that the A and B, they are in codominance with each other. Neither A nor B is dominant over each other. Therefore, when they are in combination, both of them, they are expressing themselves. Therefore, we call them as the codominance. Another important factor, another important antigen that is present on the surface of the red blood cells, that is the Rh factor. The factor which determines that your blood group is either positive or your blood group is either negative. So when you talk about the Rh factor, this Rh factor is also known as the D antigen. And this Rh factor, this was first discovered in experiments exploring the genetic relationship among animals. There were experiments going on to determine the genetic relationship among animals, and as a result of those experiments, this particular Rh factor or this D antigen, it was discovered. Now, what this, how they discovered this? What they did was that the rabbits that were inoculated with the RBCs of the rhesus monkey and the rabbits that produce a specific antibody against the RBCs of the rhesus monkey, and those antibodies also reacted against the human RBCs. Now this means that the RBCs of the rhesus monkey and the human, they are sharing something, because if the same antibody is reacting against the RBCs of the rhesus monkey and they are reacting against the RBCs of the human beings, that means we are sharing something. And that actually led to the discovery of the Rh factor or the D antigen. Now this antigen is actually termed as Rh for the rhesus monkey because it was discovered in the rhesus monkey. So if you talk about this antigen, it is present in 85% of the humans. It is present on the red blood cells of 85% of the human beings around the globe. Now if you talk about the Rh type, so a person Rh type, that results from a combination of two possible alleles. The gene for this Rh antigen that have got two alleles. One allele is dominant and the other allele that is recessive. And a person inheriting at least one of the gene will be Rh positive. Now this means that the dominant allele will express itself in the homozygous as well as in the heterozygous condition, because if a person is inheriting at least one of the Rh gene and that is making him or her positive, that means that the Rh positive is dominant over the Rh negative. Now this particular Rh factor or the D antigen that is denoted by symbol above the blood type. Like say for example, if you talk about this one, the blood type is O, but it is negative because the Rh factor is negative over here. If you talk about this one, this is AB, the type is AB, but it is positive. That means it is expressing the Rh factor. We will continue the discussion in the next video.